presented by Itumeleng Zosela, PhD candidate from the Nelson Mandela University. Her talk is entitled Plant-Based Synthesis of Gold Nanoparticles for the Next Generation of Colon Cancer Treatment.
Good morning, everyone. My name is Itumeleng Zosela, a PhD physiology student from Nelson Mandela University in South Africa. Today, I will be presenting my work titled Plant-Based Synthesis of Gold Nanoparticles for the Next Generation of Colon Cancer Treatment. Just to give a bit of background on colon cancer. Colon cancer is a type of cancer that starts in the large intestine, which is also known as the colon. Colon cancer occurs in the large intestine's glandular epithelial cells. The cancer grows when the epithelial cells acquire a set of genetic or epigenetic mutations that give them a selective advantage. It is the third most often diagnosed cancer and the fourth largest cause of cancer-related deaths worldwide. Colon cancer is usually treated with surgical removal of initial tumor, followed by chemotherapy, radiation therapy, and/or targeted therapy if the illness has progressed. Colorectal carcinomas can be classified as familial or sporadic depending on the origin of the mutation. About 15% of colon cancer cases are familial and about 90 to 95% of colon cancer cases are sporadic. Familial cancers are inherited cancers, whereas sporadic cancers are cancers associated with risk factors. The increase in colon cancer incidence and prevalence is associated with risk factors such as smoking, diabetes, obesity, alcohol abuse, high stress levels, and inflammatory bowel disease. Plant-based synthesis involves the use of plant materials as green-reducing agents for synthesis of gold nanoparticles. Current sustainability initiatives require the use of natural materials for the synthesis of gold nanoparticles because these natural materials are environmentally safe. Traditional chemical and physical techniques producing gold nanoparticles are often confronting issues of stability, particle growth control, and application. The use of plant extracts in the production of gold nanoparticles has been extensively documented to result in nanoparticles with highly regulated size and highly regulated shape. These plant-based synthesis methods are less expensive and do not require the use of harmful chemicals, high pressure, or high temperature during the synthesis procedure. The aim of this study was to investigate the use of green-synthesized gold nanoparticles from Bordetella ferruginea for potential cancer treatment. In this study, gold nanoparticles were synthesized using the stem bark extract of Bordetella ferruginea. The nanoparticles were characterized using UV spectroscopy, dynamic light scattering, Farad transform infrared spectroscopy, and high-resolution transmission electron microscopy. The in vitro anti-cancer activity of the gold nanoparticles was investigated by using the PrestoBlue assay. The Caspase 3-7 assay was used on the human colon cancer cell lines at different times to access the optimum time for cell death. The cellular uptake and localization of the gold nanoparticles was investigated by using HR-TEM. This was to assess the uptake and localization of the gold nanoparticles within the cells. Looking at the results of the study, the first thing we're going to discuss is the synthesis of the gold nanoparticles. The green synthesis of the gold nanoparticles was carried out by the reduction of aqueous gold chloride using the aqueous plant extract of Bordetella ferruginea. The gold nanoparticles were synthesized by stirring the reaction mixture at room temperature. The formation of the gold nanoparticles was detected by the change in color of the reaction mixture of gold solution and plant extract from a reddish-brown color to a deep purple color as seen in figure 1. The color change was observed within 5 minutes, which confirmed the formation of the gold nanoparticles. As discussed before in the method section, the characterization of the gold nanoparticles was done by using UV spectroscopy, FTIR, DLS, and HR-TEM. Figure 2 shows the UV spectra of the biosynthesized gold nanoparticles, showing an SPR band peak at 538 nanometer, which is in between the range of gold nanoparticles, which is expected to be between 500 nanometer and 600 nanometer, with stable nanoparticles being between 520 nanometer and 540 nanometer. Figure 3 shows the FTIR results of the plant extract and gold nanoparticles, indicating the presence of biomolecules in the reduction and capping of the gold nanoparticles. Table 1 shows the DLS results with an average size of the nanoparticles at 45.28 plus minus 3,795 nanometer, the polydispersity index PDI at 0,281, and the zeta potential at minus 20.5 MV. Figure 4 shows the HR term image and histogram of gold nanoparticles at 20 nanometer. Figure 4a shows formation of mostly spherical particles with a few triangular shaped particles, and figure 4b is a histogram of the particle distribution with an average size of 14 plus minus 3 nanometer. The next step then was to check the effects of the gold nanoparticles on cell viability. Cell viability rates are good indicators of cell health. Cell viability assays are essentially used for screening the response of the cells against a drug or a chemical agent. MTT was used to determine the inhibitory concentration at 50%, which is the IC50 value, over 24 hours. MTT measures the mitochondrial reduction enzyme's activity after 24 hours, but it is not known what happens over those 24 hours. Therefore, we did a time study over 24 hours with Presto Blue assay. The MTT can only measure the cell viability after 24-hour treatment, but with the Presto Blue assay, you can check the viability even after 30 minutes of treatment. Even though the principle of action of both assays is very much the same, the Presto Blue was used to validate the IC50 concentration obtained from the MTT results and also to see what happens to the cell viability over the 24-hour period. The Presto Blue assay was performed to investigate the optimum time treatment of colon cancer cells with gold nanoparticles. The cells were exposed to the IC50 concentration of the gold nanoparticles, a positive control which was 5-fluorouracil, and untreated cells were used as the negative control. The cells were treated at different times, which was 2 hours, 4 hours, 6 hours, 8 hours, 16 hours, and 24 hours. After each treatment, the cell viability was then assessed. Figure 5 shows the results of the Presto Blue assay, indicating the percentage of cell viability in the colon cancer cell lines after treatment with the 5-fluorouracil, the gold nanoparticles, and versus the negative control, which was the untreated cells. The negative control for this study was set at 100% cell viability. The results show 50% viability after 24 hours, which confirms the IC50 value. The results also show a consistent linear decrease of viable cells, which confirms a process of either apoptosis or inhibition of proliferation. After the cell viability assay, it was very important to check the apoptotic effects of the gold nanoparticles on the cells. Apoptosis is a crucial mode of programmed cell death, which is an active physiological process to eliminate selectively unneeded cells. The execution of apoptosis depends on caspase 3 and 7 proteolytic activity, which in turn cleaves protein substrates that cause metabolic distress to the cells. To investigate caspase activity, colon cancer cell line was treated with 5-FU and the IC50 concentration of the gold nanoparticles to determine whether the treatment promotes cell death via the caspase 3-7 pathway. As seen in Figure 6, there was significant increase of caspase 3-7 activity with treatment with both the 5-FU and the gold nanoparticles. The gold nanoparticles having a slightly more effect than the 5-FU, showing that caspase activity starts very soon after treatment. Now discussing the results from cellular uptake and localization of the gold nanoparticles. The most important characteristic that should be considered when nanoparticles are used in biomedical applications is toxicity and cellular uptake. The biocompatibility of nanoparticles is one of the most critical features for biomedical use of nanoplatforms, and the ability of nanoparticles to be internalized by the target cell is a very important target in comparison with non-target cells. In this study, HRTEM was used to determine the localization and uptake of the gold nanoparticles within the cells. A typical bright field HRTEM image of the cell gold nanoparticle complex was obtained under optimum reaction conditions. The cells were treated with the IC50 concentration for 24 hours before embedding and cutting the sample for HRTEM analysis. Figure 7a and b show features that include increased cell volume, distortion of all subcellular organelles as shown by one arrow in figure 7a and b, and blabbing of the plasma membrane. These characteristics show that apoptosis was induced in the cells after treatment with the gold nanoparticles. The plasma membrane blabbing shown by the two arrows in figure 7b could result from the caspase-mediated activation. In conclusion, the characterization techniques employed in this study revealed the successful synthesis of colloidal, mostly spherical nanoparticles. The FTIR spectrum revealed the presence of reducing and stabilizing groups in the extract responsible for gold nanoparticle synthesis. The plant extract isolated has shown to have inhibitory activity against various cancer cell lines. Even though the plant extract alone showed cytotoxic effects, the gold nanoparticles synthesized from this plant extract were more cytotoxic than the plant extract alone. The in vitro cytotoxicity studies confirmed the cytotoxicity effects of the gold nanoparticles on colon cancer cell lines, and it was determined that the optimum time treatment for IC50 concentration is 24 hours. The apoptotic assay revealed that caspase 3-7 pathway was activated in the cells treated with the gold nanoparticles, which could be through the intrinsic or extrinsic pathway of apoptosis. Although these results are promising, investigation of the specific apoptosis pathway responsible for cell death is important to understand the mechanism of cellular interactions with the nanoparticles. The uptake and localization of the gold nanoparticles was investigated using HRTEM. There was no sign of cellular uptake even though cell death via apoptosis was observed in the HRTEM micrographs. These results suggested that the activation of apoptosis could be through interaction with the plasma membrane, or it can also be ligand-activated second messenger. Further research is still needed. This study suggests that the plant-based synthesized gold nanoparticles might be a potential agent in cancer therapy. Since phytochemicals become part of the gold nanoparticle structure, the effect of phytochemicals may interact with death receptors on the cell membrane, which can activate intracellular pathways to stimulate the activation of caspase 3-7 within a few minutes. Due to lower lymphatic drainage in tumors, the gold nanoparticles may enhance the exposure time to cancer cells and therefore may target tumors more efficiently. Thank you for your time. I'd like to acknowledge my supervisors, Prof. Ru and Dr. Mufamadi, for all the support they gave me throughout this work. And I would also like to acknowledge the National Research Foundation and the Department of Science and Innovation in South Africa for funding this work.
Now it's my pleasure to introduce the next speaker. It's Professor Poudineh from the University of Waterloo. Her presentation is entitled Next Generation Enabling Technologies for Health Monitoring.
Hello, everyone. Thanks for joining me this late morning, early afternoon. So today I'm going to talk about some of the new research projects that we are working on them at Ideation Lab. So let's start. So controlling diseases and providing healthcare is one of the big challenges worldwide. So diseases such as cardiovascular disease, cancers, and diabetes and Alzheimer's disease are the leading cause of the deaths in worldwide, even in the face of worldwide pandemic that we recently faced. So we do need to test a vast number of people every day to be able to detect these diseases at the earliest stages, and after detecting the disease, we need to track and monitor the healthcare to make sure that the treatment's working effectively. So Unfortunately, most of the current technologies and current techniques for diagnostics and also tracking the health scares are like complicated and are only suitable for practicing in the main clinical laboratories. So for like if you had the misfortune of having a loved one and at the intensive care units, you know that there are like many tests that you want to do on the spot to know what's going on. And this is the same for the emergency room when we need to triage patients to be able to provide care to the ones that are at the urgent need. So we do need to have better technologies that are suitable for practicing in this type of clinical settings. But this is something that can be problematic, because currently how it works is that after that a sample is taken from the patient, it would have a long road ahead of it in terms of returning the result. So at the doctor's office or like in the like hospitals, the samples will be taken from the patient. After that, the samples should be transferred to the main clinical laboratories. And this is like where the samples usually are like they would sit for a long time, because most of these big laboratories, they do the testing when they have a bunch of samples collected. So that's the most economical way of doing testing. And after, so this doing the testing also is time consuming and requires different steps. And at the end, like after the tests are done and the results should be sent again to the doctors. Doctors should find the patient, and at the end, at this stage, an action can be taken. And this can be like several days, or like in most of the cases, several days after that the patient walks into the clinic or into the like for example, emergency room. So we do need to have better technologies, and this is like a big challenge in the healthcare. So the question is that, so this would take a few days, as I said. So the question is that, can we move testing from clinical laboratories to the point of care to do the testing at this like sort of clinical setting rather than main clinical laboratories? So think about that at the doctor's office, patient will give like kind of the samples, and on the spot, so the doctor can tell the patient what is like the problem and what should be the action to be taken. So that's the question that actually I was trying to answer like during my PhD and postdoc, and now at the ideation labs. So by developing biosensors and assays that can enable rapid and sensitive testing and like detecting of the disease. So there are a variety of biosensors exist for that they integrate different body fluids such as blood, urine, interstitial fluid, or ISF, which is a fluid underneath of the skin, saliva, tear, and sweat. So Radiation lab, we have focused on developing biosensors that for studying blood and ISF. Blood is the richest source of the biomarkers. For example, in the blood of a cancer patient, there are circulating cancer biomarkers, or in the blood of a patient who is infected with the bacteria, pathogens can be detected. And there are also several circulating protein or cancer protein or DNA that are, they can be an indicator of disease. ISF, or interstitial fluid, is the second richest, and it has the most similar molecular compositions to the blood plasma. In order to study and analyze blood and ISF at ideation labs, we are using two main technologies. One is microneedle. So we are using microneedle for ISF analysis, and we are using microfluidics for the whole blood analysis. And today I'm going to talk about some of the works that we are doing in this area. And I'm starting with our microneedle projects. Microneedles are arrays that they can penetrate through the skin, and they would enable minimally invasive access to the interstitial fluid. So these microneedles wouldn't reach the nerve. So that's why they're like, you won't feel them, and these patches will be applied into the skin. So as you can see, there are a variety of microneedles, from solid, coated, hydrogel, and hollow microneedles. At Ideation Lab, we have focused on hydrogel microneedles because of their several advantages. So first of all, they can enable increased and rapid access to the ISF, and they're also highly biocompatible, so which is very important for like if you want to do in vivo sensing. And also they can be fabricated like easily with the low cost, they have a low cost fabrications, and they can be fabricated in for high production, which are like important points when you want to like move a technology to the market. So these hydrogel microneedles, they have shown promising results for the drug delivery, and they're already in the clinical trials to seek FDA approvals. But their application in sensing is more recent, and current technologies, they only extract ISF for off-site testing and analysis. Again, it requires laboratories, and which is time consuming and also expensive. At Ideation Lab, we are working to integrate the sensing elements inside these hydrogen microneedles to enable rapid and like sensitive testing without the need for the clinical laboratories. I'm going to talk about some of these projects that we have done. So in one of these projects led by Hanjia, a great PhD student in the lab, and Amin, our collaborators, we developed a reagent-less fluorescence microneedle assay for minimally invasive detection. Hanjia is going to talk about these projects in details tomorrow, so I would highly encourage you to attend her presentations. But briefly, sorry. We have like these hydrogel microneedles. We use a novel technique to link aptamer probes into our hydrogel microneedle. So we use aptamer as a recognition element. To make it reagentless, we link a displacement strand. We link a displacement strand to the aptamer, hybridize the displacement strand. So our aptamer has a fluorescence molecules, and the displacement strand has a quencher. So we link these components into the microneedles. In the absence of the targets, there isn't any fluorescence because the fluorescence molecule is quenched by the quencher. But in the presence of the specific target, so sorry, you cannot see it here. So it was something in the problem with the presentation. But in the presence of the specific targets, the affinity of the aptamer is more toward the target, and that's why the displacement strand would be displaced, and we will see a fluorescence signal. So we don't need to apply any reagent for the detection for this technique. So here I just want to show you how this patch look like. So these are our the patch that we are fabricating it at the our lab. So they have been made using a polymer which is hyaluronic acid base, like its base is hyaluronic acid, so it's like highly biocompatible polymer that has been used also in like cosmetic application a lot. So it's already FDA approved, and you can see also our needles that they have very sharp like tips, and these sharp tips are important for skin penetration. So in this project, we use the RFMID, our approach for detection of the 4 different molecules and target biomarkers. So ATP, glucose, L-tyrosinamide, and thrombin. So again, Sid is going to talk about this project in many details, but mainly we use it also in vivo for testing the glucose in rats. And we showed that the approach is like very can closely match the current glucometer measurements. So this is like the skin rat that we applied the microneedles into them. So in another project in our microneedle basically technique, so we have developed conductive microneedles to be used for the sensing. So here we mix our polymers with the conductive polymer, which is P.PSS, and we used it as the like working electrode of an electrochemical sensor. So this is like our microneedles before and after, before insertion and after insertion, that it is swelled. So we use that in an electrochemical sensor for the detection. So in one of the projects led by Peyman, talented PhD students in the group, and I mean our collaborators, we incorporated gold nanoparticles into these conductive polymers and use it for the glucose sensing. And in another projects led by Sarah, great undergrad students who work in our lab during like her co-op program, we use the like the platform for pH sensing. So our hydrogel microneedle is dopamine based, so it is intrinsic to detect high and low pH. So in future, what we want to do to move these technologies into the next step, we want to integrate the aptamer of the RFMID with this conductive polymer and use and make platform for wearable and electrochemical monitoring, so that the patient doesn't need to like see, for example, doctor regularly. So I'm going to just very briefly talk about our microfluidic project, but just before moving, so we did like also many animal experiments here at Waterloo for testing our microneedle patches. So very briefly, I will talk about our microfluidics work. So as I said, we are using microfluidics for whole blood analysis. During my postdoc at Thompson Group at Stanford, we developed a microfluidic assay for continuous measurements of the glucose and insulin in live animals. So continuous glucose monitoring platforms have been already developed, but there isn't such a platform for continuous insulin monitoring. And that's a big, I would say, knowledge gap in diabetes research and also clinics. Because insulin dynamics is needed to be able to give patients more accurate dosage of the drugs, for example, insulin or other diabetes drugs that are that patients are using. So for this platform, we had a microfluidic device that is connected to the rat, but the assay basically we called it RT ELISA or real-time. So we miniaturized the ELISA into a microfluidic and we transferred it into a real-time platform. So we have bit-based assays for the insulin detection. So we have like the capture antibody conjugated on the surface of the beads, and we have the detection antibody conjugated to the fluorophore as well. So for the glucose, we use the similar strategy to the RFMID project. So we have the aptamer and the displacement strap. And we integrated everything into a microfluidic device that has different modules. And these different modules basically mimics different steps of the ELISA. So the main, I would say, module is the mixing module, which mimics the incubation step in ELISA. And using microfluidics, we were able to reduce this incubation time to less than 1 minute, from several hours in the ELISA to less than 1 minute for the measurements. So I won't go over its details, but I'm happy to talk later if anyone has any questions. So just wanted to show you this, that we connected the device into a rat and we withdraw blood from the rat continuously using a peristaltic pump, and we use the assay for continuous insulin and glucose measurements. So here is the results that we got. So each of these blue dots is showing the RT ELISA measurements, and the orange squares are showing like we collected blood from the tail of the rat and we did the ELISA for the insulin measurements, and we measured the glucose using a glucometer. So as you can see, The platform RT ELISA is like very well matched with the measurements that we got from the standard techniques, but the time resolution that we can get using this technique is much it's much higher. So in the next step, what we are doing, we are moving the RT ELISA to we are developing currently the next generation of RT ELISA, and we are going to add other biomarkers of the like diabetes, glucagon, and C-peptide, and we aim to do the measurements in the conscious rats, and we're collaborating with McMaster University for the next of this platform. So in another microfluidics works that Fatemeh, again, a great PhD students in the lab, led the projects with Nandini, a postdoc. So we developed a microfluidic platform for cervical cancer detection. So again, Fatemeh is going to talk about this work tomorrow. So I would definitely encourage you to attend her talk. But basically, we make a platform that can detect cervical cancer biomarkers, DNA, from a droplet of the blood. And currently, we are also developing the next generations of this platform for multiplex detection and also to improve the sensing. So that brings me to the end of my talk. So I hope that I was able to give you an overview of what we are doing in our lab in our microfluidic and microneedles works to develop assays for whole blood analysis and interstitial fluid study. So at the end, I would like to acknowledge my lab collaborators and the funding that supports our research at the Ideation Lab. Thank you very much.
Presentation. Any quick questions? Maybe we can have one question before we go to the next topic. Awesome, quick question. Just in relation to your fluorescence, I'm just curious. So after you apply, for example, onto the skin, once the fluorescence intensity kind of starts to develop, do you detect it using some sort of device while the patch is still on the skin, or do you have to remove it over a period of time? Sure.
Yeah, so for this, the first stage is a single time point measurement. So we have to remove. So for the first like setup, we use microscope for the detection, but currently we are also making a miniaturized optics for the measurements. Eventually, for the continuous measurements that we don't need to remove the patch, so we are going to make it electrochemical assay. So we are going to combine the aptamer with the conductive patch that I showed to make like conductive electrochemical assay for continuous measurements that we don't need to remove the patch.
Interesting. Thank you. Any questions from the audience? If not, we'll move on to the next speaker. Thank you so much.
Yeah, thank you so much.
So our next talk is Professor Nicholas Hunt from the University of Sydney. His presentation is on oral nanotherapeutic formulations of insulin. This will be a virtual presentation, but he may be available for a brief Q&A immediately after his presentation.
Good morning in Australia. My name is Nicholas Hunt, and I'm a lecturer at the University of Sydney with Sydney Nano and the Charles Perkins Centre. And today I'm going to be talking about our nanotechnology that we've developed for the oral delivery of insulin. So insulin is a very critical peptide that's essential for our regulation of blood glucose or blood sugar. Normally when we get an increase in blood glucose, the pancreas via beta cells releases insulin. Insulin then facilitates glucose uptake into the liver, muscles and fat to return our blood sugar to a normal level. In type 1 diabetes, there's an autoimmune destruction of beta cells that leads to the body being unable to produce insulin. And for these patients, we need to supplement their insulin intake. So most people do know a type 1 diabetic and are aware that they need to take between 4 to 6 injections of insulin each day. And these injections come with a particularly high health risk for hypoglycemic incidence. And this is when you overshoot the correction of blood glucose, resulting in low blood sugar. And this can lead to hospitalization and potentially loss of consciousness. It's been 100 years since we've discovered insulin. We've over 500 million vials sold each year, but we still don't have an oral formulation, or we don't have a safe formulation. So a formulation that reduces the risk of hypoglycemia. So the aim of this study was to develop an oral insulin formulation that had reduced risk of hypoglycemia. What we've discovered previously is that quantum dots, so materials that are between 5 to 6 nanometers in size, have a unique property when they're given orally. So these types of quantum dots are able to circulate to the liver, can have very rapid uptake quite quickly. So with these particular dots, there's up to 60% uptake in the liver after 30 minutes of ingestion. But critically, they're not systemically available. They're cleared by the liver by first-pass metabolism and are recycled back to the small intestine. And by 24 hours, over 85% of them are excreted in the feces. So we've got a very rapid delivery system that can target the liver, but not be then systemically available. And just in acute dosage studies, we didn't observe any particular toxicity of these quantum dots. If we've got this particular type of nanotechnology, we want to be able to, first of all, attach insulin to these quantum dots, but we also need to protect them from the environment they're going to pass through. And insulin's particularly sensitive to degradation from stomach acid, but also enzymatic degradation. So not only do we need to have a physical barrier, we also need to prevent its also potentially immune recognition of insulin as type 1 diabetes is an autoimmune disease. So what we've developed is a polymer of chartistan and glucose that acts as both a physical barrier, but changes the surface chemistry of these particular quantum dots that are coated in the surface of insulin to effectively make it unrecognizable as an insulin structure. The other key component of this type of technology that we wanted to be able to develop is pH-dependent aggregation. And as you can imagine, as an oral tablet passes through from the mouth to the stomach to the small intestine, it undergoes a particular exposure to low pH. Our ideal product wants to aggregate to reduce the surface area to volume ratio to limit the degradation of our product as it goes through for those lower pH environments. This is quite challenging as typically insulin, quantum dots and chartistan are all highly soluble at pH 3 to 5. So we've developed this type of technology to have a less solubility and have an aggregation property as shown here. What we have is a particular concentration of our polymer and particular ratios of our ingredients that then has a pH-dependent aggregation, as you can see at a lower pH having a much larger hydrodynamic size. This dosage is also quite critical as particular concentrations and sizing of these materials actually can induce physiological effects. So in this instance, in terms of a fasting blood glucose and also an oral insulin tolerance test. What we also want to do with this type of materials is facilitate their release of insulin in particular environments. So chartistan and glucose are broken down both by hydrolysis and also enzymatic degradation. What we observe, which is shown in red, is just under a normal hydrolysis exposure, so the release rate within water. But critically, when it's exposed to certain enzymes such as cellulase or beta-glucosidase, which have a very high concentration in the liver, we get a very rapid release, so up to 75 to 80% release within the first hour with our particular materials when they're exposed to these enzymes. So therefore, within the liver, where it needs to act, there's the right enzymes to facilitate its degradation. When we've gone through and examined this material, we first started off looking at wild-type black 6 mice and examined both the PK and PD characteristics of our material. What we observed is we had a 4% bioavailability of our materials that critically mimicked the particular Tmax onset time, or within 30 minutes, but with a much lower dosage that could potentially be used, suggesting there'll be a relationship between how much insulin we need to give and therefore more could need to be given with our particular technology. Critically, what we did observe, though, is comparisons between the liver intake between the injectable versions and our oral formulation. A similar amount was observed within the liver, both within after half an hour and also 2 hours, but interestingly, not systemically bioavailable. What we also observe with our pharmacodynamic effect is one of the key characteristics of injectable insulin is you can continue to have a dose response effect until you can induce hypoglycemia, which can be quite substantially and dangerous to particular animal models. And that's what we observed here. When we increased that dosage so much, it induced an event in our animal models that required intervention. What we observe with our quantum dot formulation of our oral insulin is it reaches a maximal effect size and then it plateaus out. And you can see down here in our high dosage graph, when we give our oral formulation of insulin, it reaches a very low level, but isn't similar to what is observed for hypoglycemia. So there's no reduction in blood sugar below 3 millimoles per litre. Whilst with very higher dosage of injectable, these mice then require interventions, otherwise they will then go on to lose consciousness. So the maximal effect size that can be contributed to from the oral formulation by itself cannot enter that dangerous dosage or dangerous event size. We then moved on to examine other types of animal models for type 1 diabetes, and the first being nod mice. So these have an autoimmune destruction of the beta cells in the pancreas. So this allows us to perform a head-to-head comparison between injectable and our oral formulation. What we observe is there is a similar effect size and similar time frame to induce effect between the injection and the oral that was reproducible both in early stage diabetic, moderate and late stage diabetics. What we also observed is more oral insulin needs to be given compared to the injectable, but we were still able to observe a dose-dependent effect. So in our PD graph here, we still get a dose-response effect with both the injectable and the oral formulation, but just the dosage used needs to be higher for the oral formulation. We then move on to a toxin-induced destruction of the pancreas with STZ-treated rats. Again, with these animal models, we observed a dose-dependent effect with a right-shifted effect size for our oral formulation. However, what we wanted to further investigate within this animal model was the effects of chronic prolonged treatment and also self-administration of our oral insulin technology. So these rats are self-ingesting their insulin in their drinking water, and during the treatment period, they could then maintain their own blood sugars within an acceptable range for this animal model, whilst the animal models that required injectable insulin had to be injected once or twice per day in order to reach their standard blood glucose level. During the washout period, our oral formulation of our insulin then restored back to the similar levels that we saw with the controls. The final key characteristic of what we observed with our oral formulation is that with injectable insulin, you have a continuing week-on-week increase in body weight within these rats. But with our oral formulation, they were able to maintain their body weight. And this effect hasn't been observed previously with other types of new insulin technology. As a final form of validation, we examined an insulin-dependent effect in non-diabetic baboons, and this experiments were performed independently by Scott Heffernan. So an insulin tolerance test means we give insulin at time 0 and then observe the effects on blood glucose. What we observed was a dose-dependent effect as we increase the dosage of our oral insulin, and critically we also observed an effect size within 15 to 30 minutes, highlighting this technology can be used to induce the same effect size as injectable insulin within the same time frame. We observed in these animal studies no incidences of hypoglycemia, and in escalating doses of treatment, we also performed biochem, lipids and hemodynamic analysis to show there was no toxicity following 4 treatments. To conclude, we've developed a drug delivery platform that allows the oral administration of insulin, but also another peptide liraglutide that can be rapidly absorbed and highly effective in managing blood glucose. We've verified this within wild-type mice and 2 models of type 1, animal models of type 1 diabetes. We've also validated its effect and its non-toxicity in a non-human primate model. I'd just like to thank the team that's helped put all this work together, particularly Victoria, Cogger, David and Glenn, and I'm more than happy to have any questions. And thank you all for listening.
Thank you very much, Nicholas. I believe he is online. Is that correct? Hello, Nicholas. Can you hear us? How are you?
Still awake. Just.
Must be very late for you, but great to see you online.
More than happy to be here. It's a nice, friendly 2:30 in the morning. But hopefully my brain's been quite active to be able to answer any questions.
Yes, so I guess we'll open up to the audience. Are there any questions from the audience? Here's your chance to interact with someone all the way in Australia.
Don't ask
Yes.
us about kangaroo.
We do have a question from the audience. Professor Ocean Shao.
Hi, Nicholas. Thank you for the presentation. I have a question on the design of your hydrogel. You have tryptophan glucose copolymer. So can you elaborate what's the rule of each block, tryptophan and glycol, this hydrogel system?
So it's a difficult kind of concept for our polymer. So what we've done is we've made a random block polymer in the end, and then we electrostatically attach it to our quantum dots. So it no longer really looks like a hydrogel per se. It more looks like a glycoprotein from how we kind of envision how it works. From what we've been able to tell, when we're making it, it looks very similar to a long cellulose polymer in a random block, and then it changes shape, I think, in a unique way to make it look like a glycoprotein. So it looks like the dot is kind of the central product, and then it's still got a branch chain that then grows off the outside of it. And we can change the thickness of those chains that are coming off, but just in terms of their kind of bulk material, not how long each of those segments are. It's just how much, I suppose, just how round and spherical they really are.
Thank you. Perfect. Any more questions from the audience? Or even virtually, I'm not sure. Maybe I can ask one last question before the lunch break. We're getting hungry here, I know. And Nicholas is probably getting sleepy. So I'm just curious, right now, how frequently do you anticipate patients to be taking this oral formulation? And have you considered maybe developing a more sustained release formulation where maybe the patient takes it maybe just once a week? I'm just kind of curious.
So we've got a platform, I suppose, that we're trying to develop with this type of technology. So it's applicable to both short-acting insulin, but also long-acting insulin as well. So if we want to put insulin glargine in, we're more than capable of doing it with this type of tech. I suppose we went with regular insulin because we could demonstrate such a very quick effect in a relatively short time frame. And also it's currently off patent, so there's going to be a lot less angry people coming along if we start using their other type of technology. But yeah, we kind of envision that this would need to be potentially still used with injectable insulin, depending on how because insulin, it's a very strict regimen of how usually it does get dosed by an endocrinologist. But there's also going to be other oral insulins that are going to come onto the market within the next 5 to 10 years. And we picture our product to be something that can be more relied upon pre-meals for patients, whilst longer acting versions that have that much higher systemic bioavailability would be more preferential kind of choice. So I think we'll work in tandem, I think, with other oral insulin products. But hopefully ours will be what everyone likes the most because of the improved safety profile compared to everybody else.
Perfect. Thank you so much, Nicholas. It's wonderful chatting with you again, and hopefully we'll get a chance to see you in person in the near future. Please join me in thanking Nicholas Hunt.
Thank you all.
Thank you.
Hello, everyone. Thank you for coming today. I'm going to present one of our recent works that has been published in Journal of Nanoscale. And it's titled as an integrated microfluidic electrochemical assay for cervical cancer detection at point of care testing. So first of all, what is cervical cancer? Cervical cancer happens in cervix, which is part of women's reproductive system. The etiological cause of cervical cancer is persistent infection with high-risk strains of the HPV virus. HPV virus, when it enters into the cells, it alters the normal cellular function and makes the cells to outgrow and eventually become cancerous. Cervical cancer is a global problem, standing as the fourth most common cancer among women. However, the burden of cervical cancer is substantially higher in low and middle-income countries, and this is mainly due to the lack of access to cervical cancer screening and treatment. Early diagnosis of cervical cancer is highly important because treating cervical cancer at early stages has 90% rate of survival and costs less than $500. However, treating the invasive cancer has only 15% rate of survival, and it costs more than $5,000. Interestingly, recent studies suggest that there are circulating nucleic acid or cDNAs that can act as great biomarkers for cancer diagnosis and also screening. These cDNAs are released from tumor cells into bloodstream. And since they are present in the blood, their sampling it can be done in a relatively non-invasive manner. And also then they can be sent for analyzing. Even more interestingly, recent studies suggest that these high-risk HPV DNAs also release cDNA into the bloodstream, which can be used as a marker for cervical cancer prognosis and also as an indicator of cervical cancer recurrence after treatment. To detect these high-risk HPV cDNAs, the blood should be collected, its plasma should be separated, and then it should be sent for being tested for high-risk HPV. The conventional methods to do all these is time-consuming, labor-intensive, and require bulky equipment. Therefore, for this type of detection, for detection of high-risk HPV cDNA to be done at remote location and in a point-of-care setting, we need a miniaturized plasma separator that is integrated with a high-risk HPV cDNA detector. Yes, so I was saying we really need an integrated device that can do both the job of separating the plasma from blood and also detecting these high-risk HPV cDNAs. That's why we designed such an integrated device. Our device has two main sections. Our device has two main sections. The first one is a microfluidic device that can take in the blood and separates the plasma and then directs it to the second module, which is an electrochemical biosensor. In the figure number A, we can see this integrated in a side view. And in the second figure, being figure number B, we can see this integrated top view. Figure number C shows the mechanism through which our electrochemical biosensor can detect these high-risk HPV cDNAs. Here we have a screen-printed electrode whose working part is covered with graphene oxide. And the graphene oxide provides those functional groups required for immobilizing a single-stranded DNA. When we have this constructed biosensor, which has screen-printed electrodes, the graphene oxide cover and those single-stranded nucleotides on top of it, we scan it in a solution that has ruthenium. Ruthenium has positive charge, which then binds to the negatively charged nucleotide in a single-stranded presented on the surface of electrode. When we scan this electrode in a certain potential in which ruthenium is reduced, those electrons are produced and then they are transferred through the conductive surface and we receive a signal. When there is high-risk HPV cDNA present in the solution, it binds to its complementary strand, which is already immobilized on the surface, and therefore there will be more nucleotide on the surface, and more ruthenium will bind to the surface, and more reduction happens, leading to production of more electrons, and more electrons get transferred through the surface, which gives us a greater signal. In figure number D, we can see signals obtained before presence of the high-risk HPV cDNA and after presence of the high-risk HPV cDNA. And as it's clear, we see a greater signal when there is high-risk HPV cDNA in the sample. Now moving to our microfluidic module. In the microfluidic module, we have three main components. The first component is a membrane that filters out all the red blood cells and white blood cells and the parts of the blood that we don't want, and takes the plasma in and passes the plasma in. We also have some parallel capillaries and capillary micropump, which together they produce a capillary force which helps the plasma to get passed through the filter and then directs it towards the outlet. We tried different shapes for the capillary micropump, and we found that the oval shape gives us the highest performance and a higher volume of plasma. We also try to invert our microfluidic, and we did this to prevent accumulation of the blood cells on the filter. When we inverted our microfluidic, because the blood cells were sedimented on the bottom, the plasma had a better reach for the filter, and therefore we could get significantly higher volume of plasma when we inverted our microfluidic. Therefore, we proceeded with all these things that we found out for the rest of experiments. Next, we move to fabricate our electrochemical biosensor. To fabricate this electrochemical biosensor, we have a screen printed electrode. Whose working electrode is, we covered it with graphene oxide. Graphene oxide, what it has carboxyl groups. And these carboxyl groups can then be activated. These carboxyl groups can then be activated to covalently bind to NH2. And this NH2, we modify our single-stranded nucleotides with an NH2 with an amine group. And this amine group then can bind to carboxyl presented on graphene oxide on the surface of our electrode. And then eventually we would have that constructed screen printed electrode, which has nucleic acid strands immobilized on it. To verify that such a covalent binding has actually happened, we performed FTIR. We did FTIR on a graphene oxide that has probe and one that doesn't have this nucleic acid probe. As this figure shows, when we have graphene oxide with probe, we see less carboxyl groups, which indicates these carboxyl groups has been engaged in that covalent binding, and the covalent bond has formed successfully. As I explained earlier, when we have high-risk HPV cDNA in our sample, we expect to see a greater signal, which has been the case here also. So when we had the double-stranded formed on the surface of electrode, we saw a greater signal. However, we did not such a signal when we had a random non-specific DNA in our sample, proving the specificity of our biosensor. Further, we obtained a very good correlation between the signal change and the concentration of high-risk HPV cDNA, indicating the accuracy of our biosensor. Now that we fabricated each of our modules individually, the passive plasma separator and the electrochemical sensor, we moved on to integrate these two together. However, before testing the plasma with our electrochemical biosensor, we wanted to make sure that our microfluidic device is capable of retaining that high-risk HPV cDNA. So we designed an experiment. In this experiment, whose data are shown in this figure, we spiked blood with different concentration of HPV cDNA that is conjugated to a fluorophore. Then we added those bloods into our microfluidic device, and we checked the plasma for presence of that fluorophore. And as this figure shows, the amount of flow for detected in the collected plasma was very similar to what it was in the control plasma. And this data shows that our microfluidic device is very well capable of retain and restore that high-risk HPV-C DNA. So next, we move to do the same experiment with our electrochemical biosensor. So we spiked blood, we collected the plasma and checked the presence of high-risk HPV-C DNA using our electrochemical biosensor. And again, we observed that our electrochemical biosensor can accurately detect these high-risk HPV-C DNAs. To conclude my presentation, here we developed an integrated microfluidic and electrochemical sensor for detection of high-risk HPV-C DNA without any need to external equipment, which are usually sophisticated and expensive. As our future direction, we would like to decrease the sensitivity of our biosensor, and also it would add a lot of value if we can detect multiple biomarkers of cervical cancer at the same time. This work would not have been possible without the great work of Dr. Nandi, who is also the first author on this paper, and Mahmoud Ayman Saleh, who was a researcher in our lab, and the direction and advice of my supervisor, Dr. Mahla Poudineh. Also, a lot of other people helped us in this project, which we are very thankful to all of them. And thank you for listening and being here.